Hello, my name is Rob Salguero-Gomez. I am an associate professor in ecology at the Department of Zoology of Oxford, and I am also a tutorial fellow in biology at Pembroke College. Some of the research that my group conducts at Oxford and at Pembroke College has to do with trying to find the mechanisms that control aging in different species across the tree of life. Research in aging is founded on the idea of the search for the fountain of eternal youth, here depicted in a painting by Lucas Cranach the Elder from 1546. The fountain of youth is a mythical spring that restores the youth of anyone who drinks from it or who bathes in its waters. Senescence, sometimes referred to as aging, is the physiological deterioration of an organism that ultimately results in a decrease in reproduction after age at maturity and an ever-increasing risk of mortality from maturity until the individual finally dies. What's important to know about aging is that it's not universal. The pattern that I described before, whereby fertility decreases with age and mortality risks can increase with age, is not found across the tree of life. Some species do have it, some species do not. And so a key critical question in the research agenda of aging research is how come that we do senesce or age, whereas some other species, both plants and some animals, do not undergo senescence. This is a really important question to ask because senescence affects most of the things that we humans do. Just think about it for a second. How long you will live, which is determined by the rates of senescence, will most likely affect how likely you are to obtain that bank loan so that you can buy a house, or perhaps that new car that you may be in desperate need of. But it will also affect you in the way of the materials that the plant kingdom and the animal kingdom provide to you. The foods, the fruits, everything that comes to your table is a function of whether the individual is able to continue to reproduce throughout its lifespan, or whether the productivity of that given species declines with age. Senescence also has got some really important aspects and ramifications for our daily lives, such as how long we work, how many days do we work, and at what age do we retire. But also it has some important implications in conservation biology and in evolutionary biology. No wonder, since senescence cuts really across so many scientific disciplines and so many dimensions of what it is to be a human being, that aging research has produced what's probably over 300 or 400 theories. In this paper by Medelev back in 1990, he was able to archive already 300 theories on why senescence or aging evolves. Hamilton, one of the three founding fathers on the classical theories of the evolution of senescence, was very well convinced on the fact that senescence was universal. So much so that he, in a peer-reviewed publication, stated, and I quote, "Senescence is inevitable. It should be found even in the farthest reaches of the universe." Now think what it means for an academic of such a high influence and prestige to say something as blunt in a peer-reviewed publication. What it meant is that he was very much sure that the patterns that you can see on your screen, that is, increases in the mortality risk after age at maturity in red, and declines in fertility after an initial peak in reproduction in blue, should be found more or less in the same shape across the tree of life. A few years ago, in 2014, my colleagues and I put together a state-of-the-art data set including dozens of plants and animal species for which we could test whether this hypothesis, this prediction by Hamilton, was in fact true or not. What we found is that both in modern human societies and in hunter-gatherers, as well as in some birds and fish like the guppies, these patterns were indeed found. So what it says is that these species do adhere to the predictions from Hamiltonian senescence. However, we have found a number of tree species, reptiles, and some mammals like the yellow-bellied marmot, where yes, the mortality does increase with age, from age at maturity in the x-axis until 99% of the cohorts dies. But interestingly, the probability of reproduction fertility does increase or even plateau with age. Similarly, we also found some brown algae, trees like oaks, and some other reptiles like desert tortoises, whereby not only did the fertility increase with age until mortality kicked in, but the risk of dying tended to decrease with age. So this highlights the needs for really the exploration of the senescence patterns across the tree of life. And one of the things that in my research group, the Salgo team at the Department of Zoology in Oxford, we've been doing is collecting data directly from the field ourselves, going to really remote places, as I will show you later, but also collecting demographic data that have been collected and made published by other researchers around the globe. What this is allowing us to do is to, for the first time, address what are the likely mechanisms for the evolution of senescence, but also for the escape of senescence across the tree of life. We have come up and tested subsequently a series of hypotheses. The first one of which was to test whether the kingdom, that is the taxonomic affiliation to which different organisms belong, thus predicts whether those species will undergo senescence or will be able to escape from it. So what you have here is different taxonomic classes of animals in blue and plants in green. And we calculated from these databases that I mentioned before, Compadre and Comadre, the rate of senescence. Species are then ranked according to whether they can escape from senescence, if the rate of senescence is higher than 1, or if they are subject to actuarial senescence, or an increase in mortality as individuals get older. And what you can see is that within each of the kingdoms, the plant and the animal kingdoms, there are some taxonomic groups, such as Magnolia diapsis, or fishy bones, or bivalves, insects, mammals, for which senescence tends to be the rule. However, you can also find some other groups, both in animals such as sponges, for instance, and reptiles, and quite a few groups in the plant kingdom, such as cycads and the taxonomic family where the pine trees are found, where species are found to most likely escape from senescence. So what this means is that taxonomic affiliation is not really a good predictor on the chances to evolve or escape from senescence, at least at the kingdom level. Work that has been led in my group by a PhD student in collaboration with a postdoc, so these are Mark Roper and Paul Capdevila, which is currently in review, has tested the hypothesis of whether phylogenies can really help us predict whether a species undergoes senescence or escapes from it. And this is pretty much based on the idea that as all species have emerged across a tree of life at different times of emergence, species that are more closely related should be more likely to find the same patterns on senescence. If this were the case, then what we would expect to see is a clustering of blues for species that escape from senescence, or yellows for species for whom senescence is neutral, that is, the mortality risk remains flat or constant with age, and species that undergo senescence in pink or reds. Those will be the species where the risk of mortality after maturity increases. In this tree of life, in this phylogeny, what you can see is in gray species of vertebrates and in pink species of invertebrates. And on the top left and right, you can find the proportion of species that escape from senescence, have got neutral senescence, or undergo senescence. And the important thing to pay attention to in this graph is the fact that there's no real clustering of pinks, yellows, and blues. So the phylogenetic signal is actually quite low in our ability to predict senescence rates in animals. Let us flip back to the plant kingdom. The way in which this graph has been structured is quite similar. The only difference is that you have got on the in the inside phylogenetic tree, you can find angiosperms, like flowering plants, in green, and then in a darker yellow, you can also find gymnosperms. So those are species like conifers, for instance. The same is the case. You can find species within angiosperms and within gymnosperms that do undergo senescence, escape from senescence, and have got neutral or flat mortality risks as individuals get older. So again, phylogenetic signal doesn't seem to be a really good predictor of whether a species in the plant kingdom escapes from senescence or not. Another hypothesis that we have been testing in the lab recently has to do with an idea that was actually put forward quite a few centuries ago. I'm going to take you back to ancient Greece, where Aristotle was the first researcher or philosopher to hypothesize that in order to live long and have a prosperous life, one should go for long walks and not eat too much. This hypothesis that Aristotle first wrote, or for which we have first records of, is nowadays commonly known as calorie restriction. In calorie restriction, individuals in different treatments who are exposed to a limited diet are shown to sometimes, but not always, live longer. This has to do with the idea that as respiration is happening in your cells, in order to be able to obtain the energy that is required to obtain from the nutrients that you intake, a reactive oxygen species are produced, unfortunately, as a byproduct. ROS, or reactive oxygen species, are nasty elements, you see, and then they can go on and bind with RNA or even with the DNA, the genetic code that is archived within the nuclei of your cells. When this happens, nothing good comes out of it for the most part unless some enzymes fix it, and that can drastically decrease the functionality and the vitality of the cells within the organismal body. Now you can think of the amount of resources that we can find around the globe as predicted by the amount of vegetation that you have. At the end of the day, most of the fauna that we see around the globe is directly or indirectly substantiated by the amount of food that plants can provide for us animals. We have obtained some preliminary evidence in the group that the extent to which any of the species that we have got access to in these databases I referred to before, so we have got access to thousands of animals and plant species, the extent to which they manage to escape from senescence is predicted by whether they live in more arid ecosystems or not. So species that are based in regions like South Africa or in some deserts in the US, as well as in Asia, have a higher propensity to live longer and to live at lower rates of senescence. Examples of species that live in really nutrient-poor environment are pretty well known to us humans and to biological researchers. Some examples have to do with bristlecone pine. Bristlecone pine is known to be the current world record Guinness of non-clonal organismal longevity. Some individuals of bristlecone pine, which are found in the west of the US, are known to have very well and easily surpassed 3,000 years of longevity. I'd like you to take a second for that to sink in. 3,000 years. What this means is that for some of these individuals, they had already become a juvenile by the time that quite a few civilizations had started to originate, including the Roman Empire, the Greek Empire, and even the Egyptian Empire. A couple of years ago in Nature, there was a paper that was reporting the record longevity of animals that at a conservative estimate of 450 years. Again, take a second for that to sink in. 450 years. This was the case of the Greenland shark. The Greenland shark lives in really nutrient-poor habitats. So beyond the anecdotal correlations between living in poor habitats and living long, we actually, for the first time in my group, have provided some preliminary evidence that for hundreds and hundreds of plant species and animal species, living in poor nutrient habitats is actually a good predictor for living long. In the previous slide, I discussed how my research group has provided some preliminary evidence for hundreds of plants and animal species showing that those organisms living in more arid or more nutrient-poor environments tend to live longer and tend to have lower rates of senescence. My group in the last years has been using demographic data for hundreds of plant species to at the same time ask how do organisms invest differentially the limited energy that we all have access to into the key building blocks of life. That's survival, growth, and reproduction. We've been asking this same question in different ways and using different tools for plants and for animals for freshwater, for marine, and for terrestrial organisms all around the globe. And the image that has emerged from that is that all organisms, regardless of their taxonomic affiliation and also regardless of where they're found, have to face a really important trade-off. You either grow fast and live short, or you grow really slowly and manage to live long. This is really important because what that means is that if you're located, unfortunately, in a nutrient-poor environment, you don't have the resources that will be necessary to grow fast. So by the same token, if you're limited in growth, there's only one outcome of that, and that is that you get to live long. This provides for the first time with a mechanistic basis that allows us to connect senescence, vital rate trade-offs, and how the environment affects senescence outcomes. Another key hypothesis that we have examined recently in my group is how the anatomy of an organism allows it to undergo senescence or to escape from it. I will give you an example in terms of the anatomical array of how most plants are organized. Plants, or most plants at least, are simply a collection of straws. You can think of them as an array of straws whose main goal is to suck up resources, both minerals and water, from below ground, take them to above ground, sometimes quite a few meters above ground in the case of tall trees like sequoias, for instance, and up in the leaves carry out photosynthesis with the help of sun and CO2, of course. Now you can think of the ability of a given module that would be a single straw to single-handedly deliver those nutrients and the water from below ground to specific parts of the canopy. What would be required for that module to act more or less independently is to be physically separated from the rest of the modules. One way in which plants may be able to do that is by being physically distanced from other straws, or perhaps if there's not many straws. One can think of what the repercussions then would be in individuals whose straws, so-called modules by us biologists, will be unable to communicate with each other. This is how we refer to modular organisms. Modular organisms are organisms where the anatomy is simply the repetition of metameres or modules, each of which carry exactly the same function with a high degree of independence. You can then think of what will happen to that glass with the straws. If I were to remove one, yes, of course we would lose the facility to be able to suck out the resources from below ground, but the rest of the architecture wouldn't be compromised. So translating this in biological terms, imagine a plant whose module number 1, say, out of 5, is trying to obtain resources below ground from a specific patch of sand where there's no water or minerals. What will happen is that module will eventually die off and it will be shredded. However, in the opposite case where everything will be integrated, that is, the modules are acting in unison and they're able to share the resources, the entire organism will suffer the hit of a lower revenue in the short energy that module can provide to the whole organism. We have recently tested with the leadership of one of the PhD students in my group, Connor Bernard, and an affiliated postdoc in my group, Aldo Compagnone, the idea that the degree of modularity, so to the left you've got plant species that are really modular, that will be that image will represent a cross-section of a trunk, for instance, and you can see clearly 5 separated modules that don't quote-unquote speak or communicate resources to each other. And on the right you've got other bauplan, other anatomical array where there's full integration, so there's a lower degree of modularity. We have accessed the anatomical information from other databases that allows us to organize plant species across this continuum on degree of modularity. And from the databases that we have access to, we have been able to obtain the rates of senescence for those species as well. What we have found is that there's a significant trend whereby species whose anatomy is more modular have a higher propensity to escape from senescence, whereby species that have a more integrated bauplan almost definitely always evolve senescence, just as we had predicted. In the same publication, we also tried to test the same hypothesis in animals. However, we did not find a signal for that. Part of that, we argue, is that it's really difficult to organize the vast variety of the animal kingdom as we can do with this simple example of straws into a single glass of water, as we can do with plants. The way in which we organize the anatomy of different animal species is according to the degree of reiteration or metamerization of different key organs in their anatomy, such as the amount of kidneys, for instance. At the left-hand side of that graph, you have organisms that have got reticulated kidneys, so those are kidneys that have got a lot of redundancy in them. And towards the right, you've got species that only have got a really vestigial and not highly functional type of kidney, or even none. And what we can find there is that actually there's not a significant signal. So anatomy does allow us to predict rates of senescence in plants, but so far not in animals. And of course, as we wander and examine life across the tree of life, from lichens anywhere around here in Oxford, including Pembroke College actually, where there's wonderful lichens on some rocks, to some desert species like Labria trentata, or to some sponges and some corals, I cannot help but think, gosh, these are really modular organisms. Perhaps the fact that we haven't really examined how long they live or whether they undergo rates of senescence that allows them to escape from it is something that it's not been studied up until now. And in my opinion, I think that it really does deserve further attention. And taking it to other kinds of organisms, insects are known to also vary in their degree of integration or modularity. In the case of ants, for instance, we know that some ant societies are heavily integrated but modular at the same time. Different kinds of ants will carry out different works, different jobs, but there will be one ant that will live for a very long time in correlation to its body mass, the queen. It's been shown that for insects, those insect species that are more colonial also get to live for longer. I think that really exciting question that we will definitely pursue in my research group in the future is how the degree of eusociality affects the rates of senescence of organisms across the tree of life. Here I want to leave you with a little bit of a repertoire of the kinds of research that my group is carrying out as we're talking, both locally and around the globe. On the top left, you can see some image of flatworms of the phylum Platyhelminthes. What you can see there is some petri dishes of flatworms. The length of the big adults will be about 0.8 centimeters, by the way, or perhaps a bit smaller. And what we are feeding them with is some calf liver. In this experiment, what we're trying to do is evaluate what happens to different species of flatworms that are endemic here to the UK as we expose them to different amounts of food, different qualities of food, but also when we make the resources available in a more unpredictable manner as is expected to happen with climate change. The added bonus of working with flatworms is that these cute slimy creatures have the ability to regenerate in some cases because they have got a lot of stem cells around them. And what they do when things get really tough is they actually subdivide in order to be able to sustain the remaining anatomies. In other research that we're currently conducting in South Africa, we're focusing on how the demography of some endemic plant species called lithops may be affected by climate change, but also how the patterns of drought and precipitation may be affecting their rates of senescence. So what you're going to see in a few seconds, if I can scroll this over a bit more, is a beautiful landscape of some image that I collected with a drone in the Karoo Desert. This is where these species live, and it really is a fantastic landscape to be able to carry out biological research and agent research. Other species that we're currently studying are corals. We've got some collaborators at the University of Leeds with whom we are building demographic models to be able to explore the degree to which modularity in corals and sponges allow them to escape from senescence. And this work is being done both in Australia and also in the south of Japan, actually. And other work that we are currently doing more locally has to do with studying the demography of parasites. If you are local to Oxford, or if you have a park or some trees nearby, I would definitely invite you to try to take a look for some mistletoes. Those green blurbs that you can actually see on the canopy once the deciduous trees shed their leaves. What's unique about mistletoes is that they are able to partly carry out the photosynthesis by themselves, but they don't actually have to invest in the creation of much anatomical tissue in order to be able to obtain the minerals and the water that they need to fix carbohydrates. And the reason for this is that they are parasites, so they just directly tap their pseudoroots, called hostoria, into the branches of a host. And the reason that we have chosen mistletoes as a key parasite species to study questions related to senescence is that all key, most prominent theories on the evolution of senescence predicts that species will undergo senescence as a consequence of trade-offs between survival, growth, and reproduction. So we're really interested in examining what will happen once that we can identify species that can bypass or cheat through those trade-offs, perhaps because they're parasites. I hope that this has provided you with a little bit of flavor for the kinds of research that my group is conducting currently at the Department of Zoology and at Pembroke College. If you're interested in any of this research, please contact me. There you can see my email and my Twitter account, as well as that of my research group. And I would like to invite you to take a look at the book that I published with some colleagues a couple of years ago on the evolution, open bracket and escape, close brackets, of senescence across the tree of life. Thank you so much for your attention.